Welcome back to the channel, everyone. Today we're going to be talking about enzyme-linked immunosorbent assay, ELISA for short. Now, the enzyme-linked immunosorbent assay is a powerful biochemical method used to detect and quantify proteins, hormones, or antibodies, and it is a very sensitive, specific tool for immunological detection. Now we're going to look at the concept and principle of how this assay works. And to understand this, we must first understand the antibody antigen interaction. Now, when you're infected with a pathogen, let's say a virus or bacteria, the pathogen has these spike like proteins on its surface called antigens. Your body recognizes these antigens and in response produces these Y-shaped polypeptides known as antibodies, which are meant to bind to those antigens in an attempt to destroy it or find a way to get rid of it. And this antibody antigen interaction is the very basis of the ELISA. Now, ELISA uses that same principle in the lab as one of these components, the antigen or the antibody, is immobilized on a solid surface, typically the walls of a microtiter plate. So here you have your antigens, which are immobilized on the surface of a plate. And thereafter, a sample containing antibodies is added. These antibodies are meant to bind to these antigens if present. And thereafter, another antibody, which is attached to an enzyme, is added. This new antibody, this secondary antibody, is meant to bind to the first antibody, and the presence of this enzyme is meant to catalyze a reaction which will change a colorless substrate to a colored product, indicating a positive reaction. Therefore, if this first antibody is absent, there would be no color change, and that would be a negative reaction. A typical ELISA setup involves the solid phase, which is usually this polystyrene micro titer plate where your antigen or antibody is to be plated upon, the antigen or antibody to be bound to the surface, the enzyme linked conjugate for the secondary antibody, and the enzyme commonly attached is the horseradish peroxidase, and a chromogenic substrate which produces color when acted upon by the enzyme. So there are two main types of ELISA, the indirect ELISA and the sandwich ELISA. The indirect ELISA detects the presence of antibodies in a sample. It's commonly used for screening viral infections like HIV and COVID-19. Here, the well is coated with the antigen, and antibodies to be measured are added to the well plate. Thereafter, the secondary antibodies that are linked to the enzyme are added. And after that, the substrate is added, and a color change shows the presence of the antibody being measured. Now, we're going to take an example using HIV screening tests. So, a patient likely having HIV virus has his or her blood taken and centrifuged. Thereafter, the blood is separated to plasma and the red blood cells. So, these antibodies are supposed to be in the plasma if present. Now, if an individual has HIV, these antibodies would have been produced in response to the antigens found on the surface of the HIV virus. Whereas if a person is HIV negative, these antibodies would have never been produced in the first place because there are no antigens meant for it to bind to. So in the lab, you have your well coated with antigens which are structurally similar to those found in the HIV virus. And thereafter, the patient's blood sample is added, if present, the antibodies would bind to the antigens, and then the secondary antibody containing the enzyme is added, and thereafter a substrate is added, and you would see a color change, confirming that that patient is HIV positive. But if the patient is HIV negative, the antibodies would never be there in the first place to even bind to the antigens. So, therefore, after washing and adding the secondary antibody, which is linked to an enzyme, those enzyme linked antibodies would have nothing to bind to. So, therefore, on addition of the colorless substrate, the solution will remain colorless and there will be no color change. That patient would be HIV negative. Now we'll go on to the second type of ELISA, the sandwich ELISA, which detects and quantifies antigens rather than antibodies. So here the monoclonal antibodies are coated on the well plate, and then the sample containing antigen is added. If the antigens are present, they would bind to the antibodies on the well plate, and thereafter the secondary antibody containing the enzyme is added, forming this sandwich. You have your enzyme linked antibody, your antigen, and another antibody before the substrate is added. If present, if these antigens were present from the beginning, there would be a place for these enzyme linked antibodies to bind to, and therefore the colorless substrate would produce color. Now, the color change as well as the intensity of the color shows the qualitative and quantitative use of ELISA assay. ELISA is extremely sensitive, capable of detecting less than a nanogram of protein, and the common enzyme attached to the secondary antibody is the horseradish peroxidase and alkaline phosphatase. And the colorless substrates that are usually used include TMB or p-nitrophenyl phosphates. These generate visible and measurable color changes that can be read spectrophotometrically with the use of a microplate reader. Now we'll go on to some applications of ELISA. Can be used in the detection of hormones like insulin and HCG. Can be used in screening of viral infections like HIV and COVID-19. Can be used in the quantification of cytokines and growth factors in research, and can also be used in safety and environmental toxin testing. Now, in summary, ELISA is an enzyme linked immunoassay that visualizes the antigen antibody binding through color producing enzyme reaction. As we have already learned, the difference between the indirect and the sandwich ELISA is in the fact that one measures the antibodies and the other measures the antigens quantitatively. And this assay has been found to be an indispensable tool in immunology and diagnostics as well as research. Now, thank you for watching. I hope you learned a thing or two about ELISA. Be sure to give this video a thumbs up, subscribe if you haven't already, and let me know in the comment section if you have any questions regarding this assay. I'll see you guys next week. Bye.